Tēnā koutou katoa, ko ingarani me piripini ke te whakapaparanga mai, ko Leicester me Bristol te whenua tupu, ko Tamaki Makaurau te kāinga, he Industry Research Liaison Manager o i New Zealand Food Safety Science and Research Centre, ko Kyla Archer toku ingoa. Tēnā koutou katoa. On behalf of the New Zealand Food Safety Science and Research Centre, I'd like to welcome you all to the March edition of the Food Science Seminar Spotlight of 2023. Which is today on climate change and zoonoses. Is New Zealand ready? Before I introduce today's speakers, I'd like to remind you of our annual symposium, which is being held on the 3rd of July in Dunedin alongside the NZFSSRC conference, the New Zealand Institute of Food Science and Technology. Registrations are now live and you can book onto both events at the same time, so we look forward to seeing you there. Today, I'm pleased to introduce Dr. Emily Vallée and Dr. Masako Wada from Massey University School of Veterinary Science to talk to us about the existing evidence related to climate and infectious animal diseases, including zoonoses in New Zealand, and discuss weaknesses and needs for preparedness. Emily is a veterinary epidemiologist who studies the effects of climate on infectious diseases and is a senior lecturer at Massey. She's conducted research funded by NPI on the effect of climate change on grazing livestock health, and this year is starting a Wellcome Trust funded project on climate change and zoonotic diseases. So congratulations on securing funding from the Wellcome Trust. Masako is a research officer at Massey, originally from Japan via the University of Edinburgh, and her PhD was on decision-making support tools for foot and mouth disease in New Zealand. Her recent work includes developing early warning systems for disease outbreaks and applications of AI tools to various epidemiological problems. Emily and Masako have been working closely together on climate change impacts on animal health since 2017. We will have a Q&A session after their talk, so if you have any questions that pop up, then please put your questions into the Q&A box at the bottom and I'll put them to our speakers for you. So I'll now hand you over to Emily and Masako.
Thanks, Kyla. So I'm just moving things around my screen. It won't be long. Here we go. Okay, tēnā koutou katoa, kui te whakapaparanga mai ke heretonga o Winahoana, he kairanga ho o I te kuninga ke pure horoa, ko Emily Vallée aho, tēnā koutou katoa. Thank you very much for the invitation to join your seminar series to talk about the climate change research we've been conducting at the Epicenter in the vet school for the past 5 years roughly. So for us, it's a very new, but also very exciting research area, and we're really looking forward to exchanging ideas. We'll start with a very, very brief look at what actually is climate change. So it's this idea that when we live our lives the way we're used to, we emit greenhouse gases, and like their names tell us, these gases trap the heat that is emitted by the Earth, which is causing long-term shifts in temperatures and notably global warming. And when the Earth becomes warmer, water evaporates faster, causing overall more rainfall. The thing is, this is not distributed equally around the globe. So whatever happens, what happens is that everywhere weather patterns are changing and will continue to do so for quite a few more years. So scientists have been developing and refining models to predict how these greenhouse gases will affect weather patterns. And to do that, they're working on different emission scenarios, depending on how good we are at reducing our emissions, from very low emissions, where we manage to reach a net zero carbon emission by around 2030, to a very high emission pattern where climate change deniers are ruling the world. On a positive note, more and more people think that we have curbed down our emissions enough to avoid this worst case scenario. But on the downside, more and more people think it is now too late to reach this best case scenario and that we cannot avoid warming of over 1.5 degrees. And this threshold that you probably heard of before of 1.5 degrees is really important because that's where we will really start seeing big disturbance of natural ecosystems. For example, we know that if we reach this 1.5 degrees warming, most coral reefs will disappear. On a local scale, climate scientists use these models to downscale them, accounting for the influence of local topography on local climates. And if you think about New Zealand, for example, we know these big mountain ranges we have in the middle of the country will create very different climate zones in the west or in the east of the country. And in New Zealand, it's NIWA who's doing that. So here we're showing some of their projections. On the left is the temperature. First row is for summer and the second row is for autumn. And on the right is the rainfall. The top row is for winter. And the bottom row is for spring. And the different columns correspond to different years for the projections. And you'll see here this RCP 4.5. That's one of these emission scenarios I was talking about. That's one of the middle road one. That's where we're trying too hard. That's prediction that they did in 2018, and I know they're working on updating them, so it may change a little bit soon. So we know that if we look at the temperature projections everywhere in the country, we're going to have, well, basically it's going to get warmer. We're going to have hotter summers, milder winters, more hot days, less cold nights. But if we look at the rain, we have a bit more regional variation. In winter, some regions will get a lot more rain. Others will have much less rain in spring. Most of the North Island will have more dry days. Coastal areas in the South Island, less. And we're also quite interested in extreme weather events, especially with what happened recently in New Zealand. The projection tell us that tropical cyclones, for example, will likely occur at the same frequency, but winds and rainfall will be much stronger. So it means they'll potentially be more damaging once they reach our coast than they used to be. And if we think about floods, for example, like we've had, and living in Hastings, I can tell you, we've had some. It's basically a combination of this effect of climate. So basically it's going to be raining more, but also our ability to adapt to it and how ready our infrastructures are to respond to that. And we've seen damages in urban areas, but it's also important to think about rural areas. We live in a country where primary industries are really important. We farm a lot of livestock. And so for us being veterinarians, an obvious question was, okay, so how is this climate change likely to affect livestock and particularly their health? And we're, of course, particularly interested in New Zealand, what's happening here and what we can tell based on what we've been observing here, because, well, we're quite different, right? Our farming systems are quite different. For example, our dairy cattle are typically grazing all year round, which is not the case in most other countries. We also have a very specific ecosystem where we have no native land mammals, only a few introduced species. So even for the diseases that are present in New Zealand, their epidemiology and transmission cycles may be slightly different from what they are overseas. And we do know that climate change affects animal diseases. The textbook historical example, probably the first evidence that climate change does have an impact on animal health is the story of blue tongue virus in Europe. So blue tongue is a viral disease of ruminants, especially sheep, that is transmitted by midges of the Culicoides genus. And in Europe until the end of the 90s, it was present in the gray areas on the map and transmitted by Culicoides semicola. And you can see their limits on the map with the blue line, that there was their limits until the end of the 90s. But by 2004, it has spread north to these pink areas on the map. And that's definitely due to an expansion of the range of the Culicoides area because their northern limit moved further north. That's that browner line here. That doesn't explain all the expansion north. There's a little bit, well, quite a bit in Eastern Europe, where they actually discovered that other Culicoides species that were already present in Europe transmit effectively the virus. And so that's one way climate change impacted blue tongue virus. The other way was when the strain BTV8 was accidentally introduced in the Netherlands in 2006. And then spread to Belgium, Germany, Luxembourg, and France. First of all, it was a surprise because, well, all surveillance efforts were obviously focusing on the south at the limits of the Imicola area. And, well, I can tell you in France at that time, definitely all blue tongue virus surveillance was on the Mediterranean because there was nothing happening in the north because that's where we thought it would come from. It would just keep spreading north. But it occurred there. And we did have, even if we didn't have Imicola there at that time, we did have other Culicoides species that could transmit it. But we thought that it would stop in winter, but it didn't happen. The winter there then at that time was relatively mild. And so transmission slowed down, but it didn't stop. So it ended up spreading quite far west and south, causing tens of thousands of livestock deaths, millions of euros of costs in production losses and control, including vaccination, which is actually quite interesting when you're a young vet graduate just out of vet school. And that's how I ended up financing my postgrad studies, by doing a lot of bluetongue vaccination. And so when we want to study the impact of climate on diseases, the most straightforward way is probably to decompose climate in a set of variables. Typically, you have, of course, temperature and rainfall, but you also have wind oscillations, so that this El Niño, La Niña patterns, altitude, vegetation cover, moisture, and of course, extreme weather events, things like droughts and floods. And there are several ways these climate variables can affect disease. First one is some pathogens can survive in the environment, and this survival is conditioned on some optimal climate conditions that usually involve things like temperature and humidity. You also have diseases that are transmitted via vectors or have a cycle that involves a wild reservoir host. And sorry, this cycle is often affected by climate. For example, you can think about development or survival of vector species that will depend on temperature and rainfall. Quite often think about mosquitoes, for example. We also know that the way we farm animals will change as climate change starts being felt. Farmers will have to adapt and also possibly do more changes as part of their mitigation strategies. And this could affect things like stocking rates or the type of feed, which in turn may affect animal health. We also know that weather, and particularly extreme weather events, will increase the stress on animals. We know, for example, that above a certain temperature humidity index, dairy cows start being susceptible to heat stress. But even without thinking about these extreme situations, the physiology of the animals may be affected enough to make them more susceptible to infectious diseases. For diseases that rely on maintenance or intermediate hosts, then these host species will also have optimal living ranges that are affected by climate, and especially things like rainfall, altitude of vegetation, and climate change will likely affect these optimal living ranges. Sorry. So all these factors are expected to cause changes in disease incidence, severity, and spatial temporal distribution of diseases. Now, ideally, if we wanted to prepare ourselves better, it would be great if we could quantify these effects. For example, if we wanted to build transmission models to assess the efficacy of control measures, or if we wanted to build risk maps. Thing is, it's really difficult to quantify the relationship between disease and climate. To do this, the best option or way we have is to have a long time series of disease cases. If you have 10, 15 years of cases, then you start having enough statistical power to look for associations with several climate variables. And ideally, you want to do that with several climate variables at a time because they interact with each other. But these time series are very rare in animal health, especially in New Zealand, or including in New Zealand, because in New Zealand, we don't have any endemic animal disease that is notifiable. Another type of data that we have, for example, laboratory diagnostic data, they're known to be prone to a lot of biases and underreporting, which can be exacerbated when, for example, we're during extreme weather events, for example, during floods. The other type of research that could be useful is experimental studies looking at thresholds. For example, studies that would look at between what ranges of temperature and humidity can leptospira survive in soil, or at what humidity does a parasite larvae develop to the next stage and become infectious. So that's the type of information that is quite useful to try to think in terms of risk and climate change. But it's usually not enough to account for the entire effect of climate on disease. And sometimes it can be difficult to even quantify the link between the presence of a pathogen in the environment and the actual number of cases that you see. So we started our journey on this climate change research in 2017 when we received funding from MPI's Sustainable Land Management and Climate Change program to look into the effect of climate change on livestock health. So we teamed up with NIWA, and we worked closely with different stakeholders representing the different livestock industries and animal health and welfare groups. For this project, we started with a long, extensive systematic review where we looked at the climate sensitivity of over 40 livestock diseases that are endemic in New Zealand, and Mycoplasma bovis, even if it's not endemic, we included in it. And we then ranked our, prioritized our diseases. We ranked them formally, accounting for several criteria, things like epidemiological criteria, like the incidence, economic criteria, for example, the production losses or the cost of prevention and control, how easy it is to control, and social and ethical aspects, for example, if it's a zoonotic disease, how much it would affect animal welfare, how much it would change the social license of farming, for example. We excluded thalaria and liver fluke, not that we have anything against these two diseases, but there were some really good recent work that were published on how climate change is likely to affect these two in New Zealand. So we decided we're just going to put them aside because the work has been pretty much done. And then finally, we reached our top 5 and we looked at these 5 more closely. So the top 5 were facial eczema, leptospirosis, mastitis, salmonellosis, and Barber's spool, which is a gastrointestinal parasite. And interestingly, only 2 of the 5 are zoonotic, and 3 are really purely livestock diseases. And it shows that even if it's not a public health problem, it would still be a major problem for the country because of the way it affects the livestock. Having a little bit of a look at the systematic review findings. So here this bar chart shows the number of articles that were identified that explore quantitatively the relationship between disease and rainfall. As you can see, among those that had a lot of findings, we have a lot of parasitic diseases, things like hemoncus, worms, have these ones have a free living stage, and we know these are likely to be affected by rainfall, so it's not surprising to find them there. We'll also have sorry, a lot of waterborne pathogens. We found here salmonella, we found here crypto, lepto is over there. And so again, not very surprising, but it's still good to keep in mind that this ranking does not fully reflect the sensitivity to rainfall. It also reflects the fact that some diseases are impacting production more, and so more research, so we end up anyway finding more articles because of that. Now, if we look at extreme weather events, most diseases didn't return any result. It's not entirely surprising because we're really focusing on quantitative association, and quite often these studies that look at extreme weather events, they will be descriptive. They're not quantitatively comparing to a baseline. In the search, we considered storms, cyclones, heat waves, droughts, and floods as extreme weather events. And again, the results are not very surprising. We found a lot of results for heat stress, and because it's a big preoccupation, especially for dairy cows. And again, we had a lot of parasitic diseases and waterborne diseases because floods are quite well studied in terms of health impact. Now, let's have a look at our little scenarios and case studies. First one is mastitis, and we conducted a qualitative scenario and a quantitative study. The qualitative scenario was based on the literature review, so mostly using evidence from overseas. And our conclusion was that the risk of mastitis would increase in winter and spring in regions where rainfall was projected to increase significantly. And then our master student Lee looked at New Zealand data and used somatic cell count from a Massey Dairy Farm 1 for about 10 years of data, and she looked at association between somatic cell count and climate variables. And here, very surprisingly, we found the opposite of what we expected, because Lee found that when the temperature, humidity index, or rainfall increase, the somatic cell count goes down. And well, that was a little bit surprising. Obviously, we expected the other way. So first of all, it's a very, very, very small effect size, but it's still statistically significant. So as quite often in research, we ended up with more questions than answers. But it shows the need for more local data. Obviously, there was just one farm, so it doesn't represent the whole of New Zealand. But we did have a little bit of thoughts as to why we saw that. One explanation was that that was a linear model, and it may be that the effect is not fully linear across the whole possible range of temperature and rainfall and humidity. It was fitting our data nicely, this linear assumption, but outside of the observed range, it may be not linear. For example, and that's quite plausible because during the observed the study period, there was no no time where the temperature humidity index reached the levels that are considered to be causing heat stress, for example. So we didn't have these situations. We also have different farming systems in New Zealand comparing to what was being conducting overseas. So we don't necessarily expect the same type of results. Being grazing extensively outdoors is quite different from being housed in humid sheds all over winter, for example. And also Massey Dairy Farm 1 is known to have a pretty well-drained soil. So it also probably has affected the relationship with soil there. Next case study, barber spore. So we did want to have a look at gastrointestinal parasites because they're quite interesting. They have 3 living stages whose survival and development to the next life stages are directly affected by the weather. They have threshold in terms of humidity and temperature, and they like warm, wet environments. Hemoncus contortus, so barber spore. It's a pretty annoying one and causes pretty big production losses, and we also had quite a lot of findings in the systematic review, so we focused on that one. And in New Zealand, the incidence is higher in the North Island than in the South Island, mostly because it's warmer in the North Island. If you are not a vet, it's a roundworm that lives in the abomasums. That's one of the stomachs of the sheep. And it causes significant lesions and bleeding, and obviously make the sheep pretty sick. So again, we had a qualitative scenario and a quantitative study. The qualitative scenario suggested that with climate change, we would move from a cycle that is limited by temperature to a cycle where drought would be the limiting factor. So it would change the seasonality. The quantitative scenario, which was done by master student Minta, she built a nice growing degree day and water budget model based on the L3 stage development conditions. Showed that once again, it's good when we have our local data and we actually put numbers on what we think is going to happen, because she showed that we will have an increase in risk of hemoncus with time and with climate change, even in regions where there is a prediction of droughts, like Hawke's Bay and Tairāwhiti, for example. We still see an increase in risk, even if drought will be more common, more frequent in these regions. A third case study is leptospirosis. This time we looked at 2 different quantitative data sets. The first one was based on the PhD study of Uni Upiana, who looked at a cross-sectional sample of dairy herds that have been vaccinated against leptospirosis for a long time, and she found actually quite a lot are still shedding leptospirosis, a different strain than what's the vaccine. And we had a look at how this data was associated with the summer condition just before the animals were sampled, and the previous 6 weeks before they were sampled at different lags. And what we found was that if the summer just before sampling was more humid, the cows were more likely to shed, but the association was pretty small. The ratio here is pretty low. And also, interestingly, there was no clear association with the weather in the previous 6 weeks before sampling. And then master student Tony Tanner had a look at the human notified lepto cases between 1999 and 2017. And she looked at association with the total rainfall at 0 to 3 months lag before notification. So these are still intermediate results. That's not the final result, so don't focus too much on the numbers. But the trends will probably remain that if we look at lepto or enterovirus combined, then you have 1 new case for every additional 8 or so centimeters of rain on the reporting month. But also the lag at which rainfall affects notifications differs based on the serovar. So there's an association of hydro notification with rainfall at all lags, Pomona at 3-month lag, and Bellum at 1-month lag. And that's quite interesting because it may signal a difference in terms of transmission pathways and epidemiology of these different serovars. If rainfall acts really as a transmission facilitator between the reservoir host and people, and it just means that people get gets infected via contact with surface water, or that water makes contacts between people and the hosts more common, then you expect a short lag, for example, 1 month. But if it's occurring later, then it's probably first increasing through another species. For example, it would flare in livestock before it reaches people. So that's, again, raising some nice questions around the epidemiology of the disease. I'm heading over to Masako now, who's going to talk about the next case study.
Thank you, Emily. So lastly, for a case study, we looked at the climate change impacts on facial eczema. Facial eczema is a livestock disease in New Zealand caused by mycotoxin produced by fungal spores. It sounds like a skin disease, but the disease affects liver and causes various production impacts and economic losses. The fungus grows in the dead pasture litter, and animals get infected when they eat the contaminated grass. It's a seasonal disease, and cases are reported only during summer and autumn. The severity of facial eczema is variable from year to year. In some seasons, the farmers get a bad hit, but in other seasons, it's less severe. The amount of spore is thought to be associated with warmer temperature and moisture or rainfall. So we obtained 7 years of spore counts data from the local vets in Waikato. We then merged this data with the newer climate observation data in the matching locations. There were 11 climate variables, including temperature and rainfall, but we also included other variables which had not been well looked at in the past studies. We considered various time lags between the climate conditions and spore counts up to 52 weeks or 1 year. This is because we don't know at which time point climate conditions affect today's spore count, whether it's yesterday's weather or last spring weather. At the end, we had over 500 variables to consider, which were highly correlated with each other. We used a machine learning approach known as a random forest model. The fitted models let us determine which variable at which time lag was important for explaining this spore count. The results were surprising for us, as we found that Earth temperature and solar radiation or sunlight from several months prior to sampling were the most significant factors in explaining spore count. Interestingly, rainfall, which we believe to be a major risk factor, was not associated with spore count at all. The model could also be used to predict future spore counts. As shown in the plot, we projected spore count in the study area over the next 80 years for 4 climate change scenarios. The top row represents the most optimistic scenario, where there is not much change in spore count, thanks Emily, over the next 80 years. But the bottom 3 rows show that future facial eczema seasons would change. It may start earlier, become more severe, and lasting longer. In the worst case scenario, the bottom one, the biggest changes are projected to occur towards 2100. So these predictions from the model are a powerful way to inform farmers, producers, and vets to make informed decisions about preparedness for future facial eczema outbreaks. Can you move to the next slide, please? Thanks. So we are extending this approach and applying various AI technologies in syndromic surveillance. This work is in collaboration with MPI and NIWA. We're building a system to forecast climate-sensitive disease outbreaks in animals using diagnostic lab submission data. The surveillance data is collected when animals get sick and farmers call vets, who then send samples to diagnostic labs. It's passive surveillance and it has limitations in sensitivity and specificity. We also don't have exact location of farms, except for the district the farms are located in. But the advantage is that the data is already collected without additional resources, and MPI has been collecting this data for over 20 years. By merging this data with climate data from NIWA that spatially matches the case location, we can develop a better understanding of climate-sensitive disease outbreaks in animals. We're now at the stage of processing the large amount of data and testing various AI algorithms to determine which ones are more suitable. We're particularly interested in zoonotic diseases highly important in New Zealand, such as leptospirosis and salmonellosis. The ultimate goal is to fully automate the system. The data will be automatically imported and merged, and the AI models will be trained daily or weekly with minimum resource use. It will be able to send alerts when certain climate conditions are met and the risk of disease outbreaks becomes higher. This will help us to take proactive measures to prevent the spread of disease and protect animals and human health. It also can provide assurance in the peacetime by monitoring the existing data and can help us to prepare for future disease outbreaks by predicting when they're likely to happen. Next. We also spent a bit of efforts talking about our findings and research. Here are a couple of posters we presented at the New Zealand Agriculture and Climate Change Conference last month, and an article that was published in the conversation and picked up by staff. We provided information about the zoonotic risks associated with floods following the cyclone in February. We think the timing is important. What often happens after major disaster is that people move on quickly and forget about it, but we think it's important to learn from these experiences and be better prepared for future events because it's evidence that they will happen again. The International Climate Change Panel reports that the frequency and intensity of some weather and climate extremes increased in the last 200 years, in particular for temperature extremes, extreme rainfall, droughts, tropical cyclones, and so on. It's likely that this trend will continue. So it's important to allocate available resources for disease prevention, surveillance, diagnostics, and control to mitigate the risks of future disease outbreaks triggered by climate change. This sounds like an additional cost, but it's in fact investment for better preparedness in the future. Next, please. A few words about what's coming next. So we are just launching a new project called CLIZOT, funded by the Wellcome Trust. We will use AI, more specifically natural language processing, to extract and compile the evidence linking climate and disease from the scientific literature. This is because, as Emily mentioned earlier, literature review is a major undertaking, and without AI, it's difficult to keep up with the new research published every day. The project aims to create a database where, for example, a dengue researcher in the States can extract modeling parameters, or a GP in Northland can look at the zoonotic risks associated with floods in Pacific Islands. We'll also keep working on scenarios and case studies to validate the database and provide useful information. We have some ideas lined up, maybe lepto in New Zealand and dengue in Pacific, but that's still a work in progress. Finally, we would like to talk about the floods. and think about the challenges that we're facing when it's still timely. So we are currently experiencing the aftermath of Cyclone Gabrielle in real time still. Based on evidence from overseas, we know that floods can increase the risks of waterborne, rodent-borne and vector-borne diseases. But here in New Zealand, there is no increased surveillance post-floods, and challenges with diagnostics and access to vet make the syndromic and lab-based surveillance data unreliable for the time being. We only have anecdotal reports from the labs, vets and farmers. For example, there's a possible increase in lepto cases in lambs in Hawke's Bay, which actually was happening before Cyclone Gabrielle due to a wet summer. An increase in undiagnosed fever in dogs in Hawke's Bay, and NZVA organizing a supply of mastitis treatments in regions impacted by Gabrielle. Farmers in Hawke's Bay were posting in social media the need for medicine for fly strike to be dropped by helicopter. These local data are scattered everywhere, and we need to formally and systematically collect these data, or they may be lost. These data would be important to get a clearer picture of what's happening during extreme weather events and to improve our resilience. And that's it from us. Thank you for listening.
Thank you. We're happy to answer the question if there are any.
Great. Thank you, Emily. Thank you, Misako. That was really interesting to hear, and a little bit scary, but the more we know, the more for it. Just at the end of your talk there, Misako, you mentioned about the need to collect some of the data because it's all anecdotal at the moment. Is happening at all?
Well, the lab data, what I mentioned for the one of the project, it may capture some of these data, but it only happens when they are the cases are reported by farmers to the vets, and vets decided to send samples, but not if they decided not to. And about other social media and things, I don't think there's any way at the moment to collect these data and store them.
Just keep an eye out and see what.
Pretty difficult in these conditions. It's a real challenge. If you think about livestock, for example, the most affected farms, they've lost fencing. So even just testing animals and treating them is the huge challenge. So even if I ideally would love to have a good surveillance system in place just after floods to see what's happening, it's really difficult to implement.
Yeah. Based in Hawke's Bay, so what was your your immediate response when the floods were happening? Like how were you affected, and how did that affect the work that you've been doing and what you were then asked to do as a result?
So the paper we put in the conversation was really proactive from us based on the discussion we've had, and I think the observations we heard from a lot of people were that we're just not ready for something of that scale. And obviously things like animal health and even public health went maybe a bit further down the list of priorities, understandably. So we proactively started trying to engage in the conversation. And then we've also been involved, for example, providing advice to the NZVA on managing leptospirosis. We've done that a little bit. And we've had discussions with MPI on maybe not for this time, but next time, how we can do it better and learn a bit better what's happening.
Yeah, I think we're all kind of in that boat as well. Sorry, did you have something to add, Misako?
No.
It's okay. You look like you're. I still haven't got any questions from the Q&A. Is anybody? don't forget to pop anything down in there if you are interested in it. I was going to ask a question which then got actually answered by your presentation. So I was going to ask if there were any indications that we're able to tell of right now from the increased infections from animals after the flooding events. And so you mentioned some of them, but are there any others that viable diseases that might also arise that could also potentially be a threat to food safety?
Part of the disease that are worrying everyone is salmonellosis. That's one of those where we've had cases increasing in the past few years, and it survives pretty well in water. So it's one of the diseases that is really worrying everyone. And then it's more waterborne pathogens, CAMP and crypto, all these things. And Hawke's Bay, obviously, CAMP, we don't like this idea very much. So yeah, it's we're just looking is just from the animal side, that we won't be seeing that in the animals. We'll see that's probably emerge in the people, and we'll have to figure out where it came from, but we won't see it emerge in the animals first.
Yeah, we are doing some work with the kiwi fruit and the pip fruit industry on like the immediate impacts and kind of understanding how that has affected their crops as well. So yeah, hopefully that we don't see it. Yeah, interesting to know. Masako, you talked about the modeling project where you brought together the data from MPI and NIWA, which is really nice to see, integrating different organizations' data together. How far through that project are you?
We are still at the beginning. It's really difficult to extract the data in the meaningful way. And, these kind of data contain a lot of noises. So, for example, we want to, look at lepto, and there are this much diagnosed cases of lepto, but there are also undiagnosed cases with presenting abortion, which, may be lepto, but it just doesn't appear in the data, So there are a lot of different ways to extract those potentially relevant data. And yeah, we do need to spend more time in those data extraction.
Yeah. So how long is the project for?
This is meant to be about a year. We meant to wrap it up this year and see how the finding from this project can move on and be used for the system, building this system.
Yeah, great, thanks. A question come through, but it says the chat is disabled. So if you've got a question, yeah, pop the questions in the Q&A box rather than the chat box. I'll just wait and see if whoever was asking that might actually have another question. Because all right, well, I think it's now a good time to wrap up. So thank you so much for your time and your expertise, Emily and Masako. Yeah, it's been a really interesting topic to hear on, and an interesting. This will be made available on our website either tomorrow or over next week. And our next seminar spotlight will be at the end of April, with an invite coming out in the next couple of weeks. Just another reminder to everybody that our annual symposium is on the 3rd of July in Dunedin. We look forward to seeing you there. So in the meantime, thank you for your attendance. Have a good rest of your day, and we will see you next time. Tēnā koutou katoa.